So, good morning, everyone. I think we'll start now. So, my name is Trevor GrandPre. I'm an assistant professor of physics. I started literally like a couple months ago. And today I'm trained in biological physics. And so today I'm going to tell you a little bit about my research, which are these liquid-like droplets that are found in cells. So I'm hoping that it'll be interactive. So if there's something along the way that doesn't make sense, you can feel free to interrupt. It's fine. So it can be engaging. And okay, so with that, we'll just start. So today I'm going to talk to you about the physics of mysterious droplets. So here is an image of a whole bunch of droplets. That you might see if you put a straw in water and blew bubbles, you might see something like this. This is not necessarily in a cell, but this is the idea or the picture that we have when we look at certain types of things that are in cells. These like little things that don't really have a membrane, but they're sort of separated by nothing. They're just being held together into these confined regions. I can't use it, sorry. Into these confined regions, into these bubbles and cells. So I'm going to tell you about this more and introduce, what this could be like in cells. So the classical view of cells, you might have seen this in in high school or sometime, where you have, for instance, the nucleus. So the nucleus we learned is the control center of the cell. There's also the mitochondria. There's the powerhouse of the cell. There's also other ones maybe less known, but the Golgi apparatus, which is for protein packaging to send proteins around the cell. There's also the lysosome, which is pretty much waste disposal of the cell. Also, there's the endoplasmic reticulum, which is for protein synthesis. So you might have heard of all these at some point, but the idea is that these are so-called organelles, or tiny organs, that have a membrane. So there's something around them that's keeping them localized in space. You can think of like a water balloon or something like that. And now our new picture of cells is that, that's more cells, more stuff. Don't look at all these names, but the idea is that this is that same cell, like a typical animal cell. And the idea is that there's little droplets of proteins or molecules that are all around the cell. They're in the cytoplasm, they're in the nucleus, they're on the membrane of the cell. And the idea is that this is analogous to those droplets that I showed you in the beginning, where it's just, there's nothing holding them together. It's literally just proteins or molecules sticking together that are localizing into a bubble or a liquid-like droplet in these cells. And so our picture now of cell biology is that these are very important. And in fact, there's more of these so-called membraneless organelles in cells than there are membraned ones. And so today I'm going to tell you about how do we view these from a physics point of view, and also biological implications for them. And so these are also very crucial because they're important for our immune system. Some of these liquid-like droplets are for our immune system, for our central nervous system, for gene regulation. So it's very common in cells, but we're just now understanding this with this new, what they do and that they're in cells. Okay, so I want to tell you more about this. So another word for those membraneless, those liquid-like droplets are so-called biomolecular condensates. So they're just, condensates is, you might have heard of condensation, for instance, when, on your glass of ice-cold water, there's liquid on the surface. So that's the idea, is that these are condensates of liquid-like condensates. So if I showed you this picture, you might say, oh, that looks like a liquid droplet, right? You probably couldn't tell the difference, if you saw it outside, on the hood of your car or something. But this is actually proteins. This is actually proteins that were reconstituted from cells and just mixed in a test tube. And this is what comes out. So this is what we mean by liquid-like droplets. When you mix these proteins together, there's interactions between them that they stick together in a way that looks like a liquid. So this is sort of the connection. But now I want to tell you a little bit about the physics of liquids, just classical liquids like water and things like that. And then we'll circle back to what that means for cells. Any questions so far? Is this good? Okay. Yeah.
So is it surface tension that's giving them the shape they have?
Yeah, exactly. Yeah. So here it's just proteins that are being put together, but there's no so-called active process or ATP consuming processes. So this is considered equilibrium, and so it would follow similar properties of equilibrium, where it's circular because of the surface tension. Yeah, minimizes the free energy. Okay. So let's just talk about classical liquids first, and then we'll see what this means for these liquid-like droplets that we see in cells. So a classical liquid, you might have seen this before, but the idea is that this is a phase diagram for a liquid. And the idea is that in between so if I were to, so out here is just a fluid, and this line is a liquid, and this one is a vapor. And the idea is if I were to have a total concentration, imagine a test tube where I mix total concentration of a liquid, or I have a total concentration of a liquid at this point at 0.3 or 0.25 grams per centimeters cubed. It doesn't matter about the units, but just imagine this is some concentration. And then the idea is that if within here, this is telling you that there will be, your total concentration would of course be 0.225, but actually the system would so-called phase separate into a liquid-like phase and a vapor-like phase, meaning that one would be over here and one would be over here. It wouldn't, and so the vapor would be here and the liquid would be here. And this is, and the liquid phase is that so-called droplet that I showed you before, and the outside would be like a vapor, or meaning there's one phase that is enriched with the liquid, and there's other ones that is depleted with the liquid. And we'll talk more about this. But the idea is that this is giving us a process, an idea of universality in physics. What I mean by that is you can look at many different types of substances like water, carbon dioxide, oxygen, ethanol, many other substances. And the idea is that if you were to, they would all fall on this curve. So this is what we mean by, this is a general principle that near this point here, which is a so-called critical point, the idea is that near this top of this curve, many substances would fall on this curve of liquid-vapor coexistence, is what it's called. But let's talk about it a little more. So, if you were to look at, boiling water, I'm sure we all boiled water on the stove before. The idea is that this is an example or demonstrating that phase diagram I just showed you. So the idea is that you're boiling water, and then at some point, the molecules will leave and go from liquid to a vapor. And this is a discontinuous transition. By discontinuous, I mean it's instantaneous. There's not like a steady transition where some molecules slowly go from a liquid to a gas. It happens instantaneously. And this is related to that phase diagram. So another way to write that phase diagram that I showed you is that if you imagine that if you had a liquid here, the idea is that as you I think this should be 1/10. Anyway, I think this should actually, okay, so the idea is that as you increase the temperature, the idea is that it should, these two should be flipped, sorry about that. So the idea is that as you increase the temperature, this thing the density will start to expand, and at some point you'll have a discontinuous jump to a vapor. Sorry about that. But the idea is that this discontinuous jump here is analogous to boiling water. And it's related to that phase diagram. This dotted line is because of that liquid-vapor coexistence. And another way to think about phase separation, I'm introducing this concept of phase separation, meaning there's just two different phases of matter. So one way is, like I said, like liquid water and vapor water. But another way to think about this is oil and water. I think, if you've probably seen this, that if you pour oil into water and you shake it up, you might get something like this, right? Where even no matter how hard you stir it, it'll never be fully mixed, right? It'll never fully mix into something that is homogeneous or like a mixture of the two. Instead, what will happen is it will, over time, form two phases. The top layer would be mostly the oil, and down here would be mostly water. And this is mainly due to the density of the two. From the thermodynamic picture, the idea is that oil would prefer to associate with other oil molecules than to associate with oil and water. That would be energetically costly. So that's the idea. But this is another example of phase separation. So if you wanted to fully get the full phase diagram, I guess just throwing this in here, is that, this is this liquid area here, where you would have liquid-vapor coexistence. And the idea is that temperature is on the y-axis, so whatever, it could be room temperature, for instance. And density, and once again, is the density of your system. Up here, we just talked about this region. And the idea is this is the full phase diagram of water. So if you go even lower in temperature, for instance, if you freeze a bottle of water, you could get to a crystal or ice. So these are usually the three phases of matter, of water. So down here, what you would get is you would get a vapor with ice. So this is the crystalline solid. And then if you were to be up here, you would get a coexistence between a fluid, which is over here, and a solid. Other than that, there's two other points here, triple point, critical point. Don't worry about that. But this is like what is considered the full phase diagram of water. Just giving you some perspective that all of this exists. But once again, we're mainly interested in this area here. Okay, so another interesting piece of physics is that liquids are described by magnets. So this is another big feature of physics, which is that there's this concept of universality. And the idea is that if I were to tell you how there's some metals like nickel, like a lot of metals, that if you were to put a magnetic field on them, the atoms in that metal will align with the magnetic field. And the idea is that how that happens is very similar to a liquid. So the idea is, I'll show you this comparison. The idea is that if for a water, for instance, you could also consider a phase diagram with pressure. So pressure could be atmospheric pressure. It's like what we are here, but it's like, if you're on the moon, the pressure in space is much lower, so close to zero. But the idea is that you can also talk about a phase diagram with pressure and temperature. The idea is that over here would be a liquid, over here would be a vapor, and this is the line coexistence curve, or the critical points of pressure and temperature. Analogously, you could also consider a magnet, which H is like some external field. So even at a zero magnetic field, you will at some point have a critical point, a temperature, meaning that as you cool down a metal, the atoms in the metal will spontaneously have a magnetic moment. They will start to have clusters that will align just because of the temperature, even when there's no external magnetic field. And how that happens is actually very similar to how liquids form. So for instance, I showed you this curve here. It's just kind of flipped sideways now, where it's vapor and a liquid in this two-phase coexistence region. So I had this flipped before, but it's very similar. The idea is that you get that same phase diagram here, where now, as you decrease temperature, some of the, usually magnetic moments, you can be For instance, in these type of models, you usually consider it can be up or down, so just two types. And the idea is that there's some coexistence where if as you go below temperature, some will be pointed up, some will be pointed down, and you get this coexistence between pointing up magnetic moments in the material and some that are pointing down. That's probably all I wanted to say about that. I know it's a lot. Do you get the connection we're trying to make here? Okay, so this is maybe some background of the physics of liquids, classical liquids, and I wanted to slowly get back to what that means about these cells. So first, let's introduce this idea of polymers. So I'm sure you all have heard of polymers. It's a lot of the clothing is probably made of polymers. But the idea is that you can think of these proteins as polymers. So I want to just talk about polymers for a second and then see then describe what that can mean for these proteins. So within a polymer, you can think of it as having so-called stickers, and these are pretty much, they stick to other stickers. And so, it's very, the name speaks for itself there. But the idea is that these are just sticking to other stickers. And then there's a linker that is connecting the stickers. So a polymer has both stickers connected by linkers. That's it. Okay, that should be at the linker. But the idea is that they can form liquids similar to water, like I just showed you. That if you were to set up a system with enough polymers, they would form something that has the density of a liquid over here, and is something that outside that has the density of a vapor. So they would have like a liquid-vapor coexistence, similar to water. And a lot of work has been focused on, for instance, how the number of stickers within a polymer could affect the density of this liquid phase. And so, for instance, if I had a polymer with 5 stickers, and I had one with 7, what you would expect is that this is the same kind of two-phase coexistence I've been showing the past few slides now. The idea is temperature, and this is concentration. And the idea is that as you CSAT is pretty much the total is the saturating concentration, meaning if you have more than that concentration, you will phase separate and form a liquid and a vapor. If you have less than that, it'll just be a vapor. Similar over here, Cn is just saying that is the concentration inside the liquid droplet. Okay, so this is the vapor, this is the liquid. And the idea is as you increase the number of stickers, this will generally increase the area of the two-phase region. So if I had 5, I might be in the green. If I had 7, it would enlarge it generally to make this liquid more dense and make the dilute phase more dilute. That's sort of like a general principle of polymers, excuse me. So the idea is now, what does this mean for amino acids? Proteins. The idea is that proteins can be thought of as polymers. But now let's just do a quick recap of what proteins are composed of. The idea is that you don't have to necessarily be able to see these, but the idea is that proteins are made of amino acids, and these are listing all 21. These are all 21 amino acids that compose proteins, meaning like every protein that you see will be some combination of these 21 amino acids, or they're just molecules. And so they're labeled, don't worry about the labeling, they're just labeled by different biochemical properties. But just showing you that this is sort of the alphabet, as you will, if you will, of how proteins are composed. Yeah.
Are proteins themselves acidic?
They can be, I think. Yeah, they can be. Some of them. Yeah, so for instance, like the orange, what is it called? The third one here. These are showing the amino acids that are acidic. So I think it's hard to see the colors on here, but I think it's these two here are acidic. So the idea is what is primarily composed of the protein will make up the property. So if it has a lot of acidic amino acids, then you would expect it to be acidic. Okay. So okay, so these are the building blocks, so they're like the alphabet of how we make proteins. And the idea is that there's a lot of work that has been done to understand how to design proteins. But just to give you some perspective, this design space of proteins is absolutely humongous. In fact, I'll just do a few back of the envelope calculations here to show you that it's actually larger than the number of atoms in our universe. That's how big this design space is. So for instance, a typical protein size could be 300 to 1,000 of those amino acids that I just showed you. It's the same 21, but some combination of them for 300 to 1,000 in a row. Here's an example. You don't have to necessarily make sense of it. A biochemist could look at this and say, oh, this is that, or, But I'm also more of a physicist myself. But the idea is you can look this up, and there's each one of these is like, this is just some protein, where each letter is a type of amino acid from that chart I gave you. And the idea is that each one of these can encode a certain type of protein, or a type of folding, or how it interacts with other proteins. And in fact, these yellow regions are so-called ordered. Everything else is so-called disordered. So I want to talk to you about these two types of, so ordered, I mean, it folds into something, secondary structures and things like this. I'll tell you about that. And I'll also mention unordered. So let's talk about this and how we can break it and how it might boil down into that polymer picture. So first, I just want to mention that if I were to design a protein for 300 amino acids, like I said, a typical size is 300. If I wanted to design one of that length, you would have 20 to the 300 possibilities. And I don't know if that sounds like a lot to you or not, but but just to give you an idea, if you were to look at the number of stars in our universe, that's only 10 to the 22. If you were to look at the atoms in our universe, that's 10 to the 80. The difference there is even the difference. So this is just an artist's picture of the atoms and stars in our universe. But the difference, if I were to look at the difference of 20 to the 300 to 20 to the 80, even that number is larger than the number of atoms in our universe. So this is just telling you that the design space is so large that we can almost design, or proteins can almost be designed to do anything with just these 21 amino acids. This is a cool thing because they're highly programmable to have many types of functions, but it's also a difficult problem because how do you understand this molecular language of proteins? Yeah.
Could any of those be illegal? In other words, you couldn't put them together like that, so there wouldn't be that many. If you threw out these can't go together and these can't go together, don't throw any out.
There is definitely some rule, probably the design space is not independent, like each one is independent. Definitely there's some blocks of amino acids that have meaning. So that's why certain blocks of amino acids will form.
So that could be cut way down. The 20 to 300 could be cut way
potentially, down. Potentially, yeah. I mean, I think it'll still be quite high, but yes. I think another thing to mention here, maybe I'll come back to this, but another thing to mention here is that, generative AI, large language models is a big thing right now, right? And a lot of it is trying to understand this, but the idea is that there's not really even enough samples to learn how to do this because that's such a large number that you couldn't even exhaust all of the type of possibilities to learn this language, even with these large language models. There's a lot of people here working on that, trying to understand how to predict the sequences, and this is part of my research as well. But the idea is, okay, we do have some understanding, so let's see how we can boil this down into a polymer, okay? That can help us understand how they form these droplets in cells. So first, I want to distinguish between ordered and disordered regions. So ordered regions mean they're folded. They will fold into some structure. Some typical structures that you would see in a protein are, for instance, alpha helices. So these are these coils here. So there are some blocks of amino acids, maybe like 20 amino acids or so, that would form a alpha helix. This is known, and so a biochemist could look at a sequence and say, excuse me, can look at a sequence and say this forms alpha helix, even without looking at the structure or doing any biochemistry or X-ray crystallography or anything like that. Another one is called beta sheets. So beta sheets are this type of structure. And so these are two of the common ordered regions or folded regions that happen in proteins. But there's also so-called unfolded regions or intrinsically disordered. And I want to tell you about this because this is a big component of these liquid-like droplets that form in cells. So this is just sort of just showing you a demonstration. Like this is one particular sequence of amino acids. Each letter is a specific one, like G is like glycine, so forth. And this is another one that would form. So this would form a beta sheet. This would form a alpha helix. So there's another type of protein. So in those sequences, some will encode for these ordered regions or folded domains, and others will be so-called intrinsically disordered. So I want to tell you about that now because this is a big component. So the idea is that over 30% of our genome actually encodes for so-called intrinsically disordered, meaning that they do not fold into a specific structure like the ones I mentioned. And so that makes it interesting, but also very difficult to study. So for instance, if you were to plug a protein into AlphaFold, for instance, it would give you something like this, where maybe a protein would be like this, where these are these alpha helices, and everywhere where you see this red line, you would see a line like a squiggly line in these structures. That's actually the intrinsically disordered regions or the unfolded regions. And they don't take a specific configuration, so they're very hard to visualize. And so this is actually a extrapolation of the intrinsically disordered regions. And I want to tell you why they're hard to visualize and why they're hard to study, because they actually make up these proteins that form the liquid-like droplets. So the idea is that they don't actually take a specific configuration. So if I were to, in X-ray crystallography, this is how you would visualize structures like this. And the idea is that they would make a crystal out of this protein and they would shoot X-rays at it to look at the structure. This works very well for these folded domains, like the alpha helix, but it doesn't work for the disordered regions because they don't sit, the intrinsically disordered regions do not take on one configuration, but in fact, they're dynamic. So if you were able to zoom into a protein while it was in a solution, it would be, these intrinsically disordered regions would be flapping all around. They would take on a distribution of different configurations. And so every time you tried to make a crystal out of it, it would be in a different configuration. And so so actually it does not show up in X-ray crystallography. Okay. So as you know, AlphaFold actually learned is actually learned on these crystal structures. And so because of that, AlphaFold is really good at predicting folded domains, not very good at predicting those unfolded regions. And this is actually sort of the state of the art of protein dynamics, is what happens to those disordered regions. And I didn't mean to go back. So so I just said this, and so it does not show up in the crystal structures. Good. Okay. So now that was sort of a primer on what is the building blocks of these proteins and the different types of, there's disordered and ordered regions of these proteins. But now I want to describe how you can talk to them describe them as polymers. Okay. Any questions so far? Okay. Going backwards again. Okay. So if we bring back the same polymer or protein, I didn't mention these, but these yellow regions are actually the folded domains, and everything that's not highlighted is unfolded or disordered. And so so this is actually a protein that forms these so-called liquid-like droplets. And the yellow regions are actually the stickers. And this is this is a condensate that I will tell you about, or a liquid-like droplet that's found in algae that's all around the world. I'll introduce this a little bit more, but the idea is that this is a protein that forms these liquid-like droplets in algae, and these yellow regions are actually these sticker regions, and the unfolded or disordered regions actually form the so-called linkers that connect these stickers. This is a sort of the physics of what we think the interactions are for this protein and how it will form a condensate. So these will stick to other stickers, and this linker just connects the stickers. These intrinsically disordered linkers or regions form these linkers. And then if you were to mix these together, then this would, it's that beginning photo I showed you of the liquid-like droplets where you thought, it looked like water. This is, one of those that forms that. Okay. So this is how proteins can be thought of as polymers. And now I want to introduce a specific type of, I think I told you about the physics, I've told you about, in general, a lot of things. Now I'm going to be more specific and talk to you about a specific one that I have studied in the past, one of these liquid-like droplets. So here is one, so there's something called a pyrenoid. It's not paranoid, but it's hard to, I'm still not sure how to say it. Maybe it's pyrenoid, I don't know. But the idea is that this is one that I've studied in my postdoc, and this is found in something called Chlamydomonas reinhardti. So the scientific name is not important. It's just a green algae. They're found in marshy lands all over the world. And if you were to collect some, they're probably even, I'm sure they're around here. And if you were to collect some, this is them. I don't know if this actually, okay, it is, okay, good, okay. So this is them swimming around, so they actually are motile. And if you were to, I don't know how you say, fix them, as they say, okay, we'll get to that in a second. The idea is that, why is this important? So actually, you might say that it's known that trees and forests are very important for the carbon cycle, that they fix a lot of carbon dioxide. They take the carbon out of carbon dioxide as a part of photosynthesis, and they're a major player in that. We know this. And but in fact, that's only about 55% of the carbon fixation. 30% is actually done by algae. So that's a big part of our ecosystem. Is that?
What?
Huh?
50% is what?
Say it again.
Say what you just said. 50% is what?
Oh, like 55% is like forest.
Oh, okay.
Forest that they take in the carbon dioxide and release oxygen. 30% is actually done by algae. So there's a lot of algae, but that's still a very big, 30% is a large number. And so this is why it's important to study. One way they're able to do this is because they're very efficient at photosynthesis, even more efficient than plants, in fact. And so here is that same algae. This is a crystal structure. So they've taken one of these bacteria and put it in, and shot X-rays through it, sliced it and shot X-rays through it. And the idea is that here's the chloroplast. Usually in images they show chloroplast as green, and because of the chlorophyll. This would in fact be green, but the X-ray doesn't come out with color, so they kind of like color it. So the authors chose purple for some reason. Okay. And then this pyrenoid is right there. So this is one of those liquid-like droplets that are found in algae. So the pyrenoid I want to talk to you about now for a few slides now, and introduce what this thing is and how it falls into this category of these liquid-like droplets. Okay. So if you were to zoom in on this pyrenoid, you would see something like this, where this, I guess this is turquoise, is the so-called pyrenoid matrix. So that's the liquid-like droplet. There's also tubules that run through it. They're like little straws that are transporting the carbon dioxide into this pyrenoid. And as I mentioned, they're mainly in the water. And so, in fact, they're they also have an aqueous form, or one that's in water is another form of carbon dioxide. It's called bicarbonate. Okay, but the idea is that this pyrenoid matrix here is what we want to study. This is the liquid-like droplets, or condensation as I call it, is driven by two well-known proteins. Meaning if you mix those two together, you'll see those liquid-like droplets like I showed you in the beginning. One of them is rubisco. You might have heard of rubisco, maybe you haven't, but in fact, rubisco is thought of as the most abundant enzyme on Earth because like 97% of organisms that do photosynthesis have rubisco. That includes plants, algae, some bacteria. They have this molecule. And this molecule is actually the carbon-fixing enzyme. It actually takes the carbon dioxide and it splits the molecule, CO2. It splits the carbon from CO2. And that's how, that's part of, and turns it into glucose and oxygen, and glucose. And so this is this step here. And the idea is that it combines that protein sequence I showed you before with the yellow folded regions is called EPIC1. It's a acronym. Rubisco is also an acronym, but EPIC1 is an acronym. But the idea is that this will stick to this and it will form that liquid-like droplet, if you did it in a test tube. And the idea is that this is actually a crystal structure. So you can find, so rubisco is a rigid molecule, and so you can make crystal structures, and you can look at it, and you can see all of the protein. Yeah.
Would it be possible to take rubisco and copy it, or like if it's asexual or something like that? Could we use that and create more of them to a point of where we wouldn't need trees or algae anymore if we just had that molecule?
That's a good point. I think there are companies that are trying to do this. There are companies who are trying to, carbon sequestration in the atmosphere, global warming, all these things. They're trying to, design or develop technologies using rubisco where you have large, vats of rubisco that can fix the carbon. That's conceivable. I think it's still sort of a work in progress. But you bring up a really good point, actually, is that rubisco by itself is, of course, the most abundant carbon-fixing enzyme, but it's also inefficient at its job. And this is where you have the dilemma. So, in fact, rubisco is only at its maximum, about 50% efficient, which, may sound good or bad to you, but, you would imagine it that if this is the major player, that maybe it'd be closer to like 90 or 100. I don't know. But the reason is, so this is actually very inefficient. And different organisms have developed ways to overcome this inefficiency. And algae have overcome this inefficiency by forming this liquid-like droplet. And so this is an interesting example where these liquid-like droplets are absolutely necessary for biological function, but we can also look at them from a physics lens. That's that sequence again. And I think I would also mention that you might say, well, why are algae more efficient? So plants do not have this organelle, or do not have this pyrenoid. They just have what's called a stroma, and it's just a big, like a water balloon filled with rubisco. But they also have pretty high concentrations of rubisco, but it's just not a liquid-like droplet. And yeah.
Two questions real quickly. What are the relative molecular weights of rubisco and EPIC1, and what level of efficiency does that combination cause the CO2 fixation to arise to?
Mm-hmm. You're saying does the weight affect the process of the carbon fixing?
And I'm saying there's a causation, just want to get an order of magnitude to know what we're talking about here.
I'm actually not sure their weight, but okay, rubisco is of course much heavier than EPIC1, but I forget the molecular weights. I can look it up for you. Yeah, sorry about that.
Yeah, along the same lines then, do you know what the size of the pyrenoid is, the diameter?
So the whole cell, I think this should be around maybe 100, or about 100 microns, micrometers or so. Yeah,
That big? Okay.
maybe 50 to 100, I think. Yeah, because one single Rubisco, I should mention this. One single Rubisco is 10 nanometers, and but then there's many of them here. It's packed. So yeah, maybe an estimate. Yeah, let's say 20 to 50 microns. Yeah. If I'm wrong before this, I'll correct myself. Yeah. Yeah.
I've heard a lot about seaweed. Is that related to
Oh, yeah.
this?
Yeah, yeah.
The algae is the seaweed.
Yeah. Yes. Yeah. Exactly. So not all algae is motile like this one. There's, for instance, like chlorella, another algae that is more like planted. Yeah. And so that would also fall under algae. Yeah. Yeah. Okay. Oh, where's going to go with this? Oh, also evolutionarily, in the grand scheme of things, first, it's thought that first there was bacteria, and at some point bacteria became able to do photosynthesis, and then this is so-called cyanobacteria, and then there was algae. Then algae emerged at some point, maybe I don't know, like 600 million years ago or something like that. And then, so they actually came before plants, and at that time, there was not a lot of carbon dioxide in the atmosphere. There's more oxygen than carbon dioxide. So for evolutionarily, there was they had to survive. This is one mechanism that developed was introduced to increase the rate of carbon fixation, because at that time they needed it to survive. And then later on, there was more carbon dioxide in the atmosphere, and so plants did not need to have this liquid-like droplet to function because there was enough carbon dioxide in the atmosphere. Okay. Okay. So this is just showing you what it looks like when the two interact. This epic one. This is actually from a electron microscope image where these regions here are the ordered regions, or the stickers. If you kind of see they're elongated, they actually do form alpha helices. And then the stickers form alpha helices, and then this linker region is just interpolated. You can't see it in an electron microscope image, but they just interpolate it, or guess. And once again, this is actually the system. Oh, okay. This system actually, when you mix them together, will form that droplet, that liquid-like droplet that I showed you in the beginning, when you mix Rubisco and Epic one. And one application of this is that if you were to engineer the pyrenoid into land plants, it's thought that you could increase the rate of, or the yields of plants, theoretically, because they haven't been done, theoretically up to 30 to 40%. So this is how efficient algae are at photosynthesis, that if you put them in plants, you could increase yields of 30 to 40%. And so yes, there is a lot of application in, as well as the carbon sequestration, as we mentioned. Okay, so I want to tell you a little bit about this one as one of these condensates. Let's see. Another thing to mention is these tubules. Right. So these tubules, as I mentioned, they actively transport carbon dioxide into this pyrenoid matrix, which is this liquid-like droplet, so that the Rubisco can fix it. So you can think of this picture as little straws and little proteins that are binding to the pyrenoid matrix here, keeping them connected. And these tubules are important for many things, besides carbon dioxide. Transporting the carbon dioxide, they can also help in the self-assembly. So for instance, algae undergo cell division, so everything breaks down, and then it has to reform into two cells. And so it's always the case that there's only one pyrenoid, and it's always in the center of the chloroplast. How is that? Well, that's still an unknown question, but it's thought that these tubules that are traversing this pyrenoid matrix are actually part of that. And some of my colleagues have been trying to understand these like proteins that bind the tubules to the liquid-like droplet, these little things I'm showing here. And so they're starting to identify these in algae, these so-called tethers that are interacting the tubules with the liquid-like droplet. And this picture, from a theory point of view, we've been developing a way to understand how these liquid-like droplets interact with membranes by looking at a classical physics problem. So this is, you might have seen this at some point, but this is an image of a liquid that is on a surface. So this is like, a drop of water on a surface, for instance. But instead, imagine that there's these surfactants or tethers that are moving around on this surface. Because it's thought that membranes can be very fluid, so there's lipids and things that can be moved around, and so these surfactants are kind of in the membrane, they're moving around, and they can move lipids around as well. So they're mobile. And the idea is that it's thought that in this pyrenoid, the proteins in the pyrenoid matrix interact with that tubule by these surfactants. And so this is a theory that we've been developing to understand how this interaction works, and how you can get these condensate membrane or liquid-like droplet. I'm using condensate and liquid-like droplet interchangeably, but how you get these condensate membrane interactions. So traditionally, how you would think about this, so it's called wetting when a liquid is attracted to a surface. And how you would traditionally think of this is through so-called Young-Dupree equation. So what that is it's telling you that if you were to calculate all the forces that this liquid is exerting on the between the liquid and the some surface here, then you can boil all of those forces down into a simple equation, which is so-called mechanical equilibrium, meaning all the forces will balance so that it doesn't move. And that's given by this equation. So this is the Young-Dupree equation. So what this is telling you is that you can characterize the forces by this contact angle. So this liquid droplet here will have a contact angle. So what this is that if you were to just, at the very edge, if you were to just draw a line at the edge of it, and what that makes with the surface, that's the contact angle. And that's, by knowing that angle, you can know the three forces. So one of the forces is the sigma SG. So that would be the force, so-called surface tension, between the solid and the gas, or the outside the droplet. And the SL is the surface tension, or force per area, of the solid and the liquid. And the sigma LG is the force per area, or the surface tension, of the liquid and the gas. So by knowing all of these, you can relate it to cosine theta. So this is like the classical picture, where there's no surfactants, it's just literally a liquid on a surface, and you can understand these forces between the two with this equation. But as I mentioned, these tubules are interacting with the liquid-like droplet, or the condensate, via these tethers or surfactants. So one thing that we've been working on is how do you describe this equation, like this type of phenomena, within this like Young-Dupree framework. Does that make sense? Am I defining this well? Okay.
Have they figured it out?
Yeah. So actually, yeah. So there is an equation here that I don't think I actually showed the equation. Actually, sorry. But the idea is that yes, you can get this cosine, the same equation, but in fact, the surface tensions that you get, the sigma SL, has an extra piece to it that will depend on the properties of the tethers. So like how the density of the tethers that are, for instance, underneath the droplet. And actually, I'll show you some results from this. So there's two main parameters. So this is a phase diagram. This is a phase diagram of this wetting process. And there's two main parameters. One is chi. Chi, and it's energy, so it's KT is the energy scale, but just ignore that. We can set that equal to 1. This is physics. No, I'm just playing. But chi is the interaction strength of the tethers with the liquid-like droplet. And so the idea is that we're trying to understand this phase diagram between chi, this tether to liquid interaction, and rho, which is the total density of the tethers on the surface on the surface. Yeah. Okay. So these two parameters, we can characterize a phase diagram of when you will get the liquid droplet to interact with the surface and when it won't. So for instance, in yellow here, when it's 1, this is the value of cosine of theta. So cosine of theta when it's 1 means that it will wet. It will interact with the surface. So that will fully coat the surface or wet the surface. And then as you go to blue, blue is not wetting. So it means that there's not enough density of tethers, rho, or the interaction between these tethers and the droplet are not enough, chi, that they will interact. So you have to have some sweet spot between chi, so that the interaction strength has to be high enough, and the number of tethers on that surface has to be large enough to see this liquid-like droplet interact with the surface via these tethers. And this is what we think is happening in the pyrenoid. This is kind of like a idealized case, and this is actually a simulation of. We developed a theory and we ran simulations to actually see what this theory would be like. And so here, the yellow outline, the phi nucleus here, that is the droplet. And since it's radially symmetric, we could just cut it in half and look at one side. So it's meaning if I looked at the other side, it would be exactly the same, so we don't have to look at the other side. And so phi bulk is the outside this liquid-like droplet. And as you can see, it looks spherical as we would suggest, but we're not enforcing it to be spherical. This is just, equations of motion for interacting with a surface. And then underneath it, you see rho T and rho naught. So rho T is actually the density of tethers that is right under that liquid droplet that is enriched under that droplet because it's interacting with the liquid droplet. Rho naught is outside of the droplet, and so you see that it jumps up from rho naught to rho T. That's because the tethers are being enriched right underneath the droplet. So as I mentioned, this is just we're actually this is actually a paper in review. We've recently updated it like in the past few weeks, but this is sort of a theory to help understand how the pyrenoid matrix could form and interact. How the pyrenoid matrix could form with the tubules via these tethers. Okay, so from that, I also wanted to mention a few more things. That was just one example. There's many other examples of droplets, but I thought I would just show you one. The idea is that I've talked to you about so far how liquid droplets are good. They're important. But in fact, there's a lot of droplets that are not good and you don't want them. This is actually a list of potential diseases that are linked to condensates. So for instance, cancer. There's some condensates that when they go awry could lead to metastasis in cancer. There's some that are related to diabetes, and there's some that are related to neurodegenerative diseases. In fact, there's some viruses that will use condensates or form liquid-like droplets in cells to evade our immune system. But today I want to just mention how neurodegenerative diseases are linked to these liquid-like droplets that should not be there. Okay, so I'm making a transition from good droplets to bad ones. So one is related to neurodegenerative diseases. We know, of course, that they can irreversibly steal the most essential human functions, such as the ability to walk, to remember, to breathe. It's sad, right? But there's actually a lot of effort to try to understand how to stop these diseases through preventing these liquid droplets from forming. So for instance, we know, I mean, not that I need to tell you too much, but the idea is that neurodegeneration is a growing global emergency. You know, it's not known that over 50 million people suffer from neurodegenerative diseases such as ALS or Lou Gehrig's disease, also known as Lou Gehrig's disease, Alzheimer's, Parkinson's, and so forth. And it's projected to increase to 78 million by 2050, not because there's more cases, but because our ability to diagnose them will improve. And so there's actually more people than 50 million now that have these diseases. And of course, the deaths related to those diseases will also rise due to accounting of the diseases. And there's no cure. And so it's thought that a common theme across these diseases, such as ALS, which I'll tell you about now, is that they form these, what I'm calling as toxic tangles, or these liquid-like droplets in cells. So here's a neuron cell. And if you were to look close to the nucleus, there's these little red this is a cartoon, little red droplets, right? Those here are toxic. You know, they shouldn't be there. So over time, this is like clumps of proteins that have gone awry, and as they grow, you know, on the right, you can get degeneration, where these droplets are so big and it just causes the neurons to degenerate. And so for instance, in ALS, I'll just give you an example. So in ALS, it's thought that there's one protein, it's called TDP43, you don't have to know the name, but the idea is that this is one where it's been pretty well linked to this disease, meaning that if it forms these droplets, it's bad, and it seems to be directly linked to this disease. But when it's well functioning, TDP43 is important for gene regulation in normal cellular physiology. So it is important to have these proteins in our cells. Once again, there's no cure, and there are FDA-approved drugs that can only slow the symptoms, but they not cure it. But once again, the idea is that there's a lot of work and effort being done to design peptides or short protein fragments or other methods to prevent these liquid-like droplets from forming in cells. The idea is with the short protein fragments, maybe they will interact with the protein in such a way that it will change the thermodynamics or the formation of this liquid-like droplet and cause it to melt or, you know, to disappear. So this is an active area of research. There's a lot of money being thrown into this, as well as a lot of academic endeavors and as well as like company startups who are trying to understand how you can so-called drug these droplets to prevent, to cure diseases. Okay, so with that, I just want to give you a little summary. So there are many liquid-like droplets containing proteins within cells. I told you about one main one that is in algae, is the pyrenoid. We're still learning the molecular language of proteins, which has a huge design space, potentially much larger than the number of atoms in our universe. And there's also some droplets are necessary for healthy cells. I haven't told you one's necessary in human cells, but there's many. I showed you a diagram of many droplets in cells. Many of them we need. If you didn't have them, we wouldn't, you know, they need to be there for us. And there's others that are bad for us. So I just mentioned the TDP43 droplet that forms within neuron cells that is related to ALS. So hopefully you enjoyed this, the physics of these droplets within cells, and this is the end of my talk.